Yeah, you can go.
Good evening, everyone. Myself, Sriharan. I'm a PharmD graduate. I'm today going to speak on clinical research in personalized medicine. It is one of the important and exciting topic in clinical research. We will be go on it. The personalized medicine. The personalized medicine is nothing but it will tailor the medical treatment to individual characteristic of each person. If, for example, I'm saying if we go to the shop, a cloth shop or shopping for any dresses, there should be no one dress fit for all. We have an individual dress depending upon our weight and size. That way we will make a dress to us. Likewise, the personalized medicine is also for personalized for each person. It should be each person. It not for one size fit for all approach. It should transfer the one size fit for all approach into individual approach. The treatment based upon the individual patient, depending upon their genetic testing. Based upon their genetic testing, the personalized medicines can be delivered. In personalized medicine, you want to remember triple R. You all know about triple R movie. Likewise, you want to know about triple R. The what main triple R thing is the right track, right time, right person. Select the right track and given to the right timing, and the right person receive that track is very effective for them. That so likewise, our personalized medicine should be presentation should be takes place deep on that. I will go move on to that. Here I'm say the example for one side does not fit for all. Why I'm saying means if we taking some drug, if we taking some drug means it will effective for some patient, ineffective now. It also now produce no effect for some patient, and also the drug produce adverse drug reaction for also some patient. Likewise, this is the example for one size does not fit for all. Sorry, one size does not fill for example, if antidepressant drug, the 38 percentage of the drug should be effective to 38 percentage of the people. Some people receive ineffective, there is no effective. Some people receive adverse drug reaction also. So this is the percentage of the drug. One size is not fit for all approach. It should be individually takes place. While we taking the individually approach means it will gain the patient outcome very quickly recover. It takes place very quickly recover. The second picture in my slide will be taken from while I'm receiving the fourth slide. I will back to this slide. I am saying the individual about individualizing drug in personalized medicine. While we adapting the personalized medicine, there are the 6 category stages that takes place. First, risk assessment. The risk assessment is done through the genetic test. The genetic test reveal what are the predisposing character. The predisposing character is nothing but any genetic characteristic or environmental characteristic or lifestyle any modification takes place that will affect the disease should be revealed through the genetic testing. After genetic test, prevention. If you want to remember this means, I will tell in one example. We go to the hospital mean, first of all, they will examine us. They will examine us. After examine, they will give diagnosis for us. After diagnosis, they will move on to the treatment. Depending upon the diagnosis, the treatment takes place. Likewise, in personalized medicine, first thing is assessment, risk assessment. After the risk assessment, the preventive cares and things that takes place. The preventing care how that prevent the disease mean any lifestyle modification, behavior changes, any treatment intervention or treatment modification to prevent the disease that takes place. And also, while we adopting the personalized medicine, early detection of disease should be takes place. For example, if the patient have gene expression variation in BRCA1 breast cancer gene 1 means the patient have 82% of occurrence of breast cancer. While we adapting the personalized medicine, that early detection of the disease also detect, and we also prevent the disease very quickly. Then afterwards diagnosis. The individual drug personalized medicine will help to diagnosis accurate diagnosis. There is no trial error. It gives targeted therapy to accurate diagnosis to the treatment. Then treatment. Major personalized medicine through the targeted treatment achieved a high range in clinical outcome, and it also reduced the side effect also. And the last thing is management. These are the things that takes place. Actively monitor what are the things are have adapted in this personalized medicine should be actively monitored throughout the person can be recovered. I'm move to this slide. This is without personalized medicine. Likewise, some may benefit, some do not may benefit. This is the patient, this is therapy. Some patient may be benefit, some patients are not do not benefit. Some patient experience any side effect. While adapting from this characteristic, from this above all this characteristic, while adapting personalized medicine through the biomarker diagnostics test, the therapy should be have each patient benefit, not should be one side fit for all. When now we move to the benefit, shifting emphasis in medicine from reaction to prevention. Likewise, before I'm saying that if the person having the woman experience having a BRCA1, a breast cancer gene expression have gene variation takes place or overexpression takes place, means they have a chance of coming 82% of breast cancer. Through this method, it help to reaction time is less and preventing time is high. While adapting this, reaction time is less and preventing time is high. And also, while adapting the personalized medicine, targeted therapy given and reduce the trial and error prescription. What means if the breast cancer patient having means that should be a two types, HER2 positive and another one is ER positive patient. While we targeting the disease, these patients are ER positive. These patients are HER2 positive, HER2 positive based upon their genetic testing. It gives target therapy, and there is no there is no trial and error. If we start empirically, it won't work means we will change that. It won't work means we will change that. There should be a long process, and it also trial and error prescription. So through this benefit personalized medicine, trial and error, it reduces the trial and error prescription, and also main thing, it will also reduce the adverse drug reaction, because the majority of the hospital in 100%, 30% of the adverse drug reaction has been takes place. Some are unnoted, some are unnotified, some are unfiled. So that is the main thing. It will reduce the adverse drug reaction and increase the patient adherence to the treatment. What adherence to the treatment means? If the treatment going well, it receives the good effective to the patient means automatically the patient will adherence to the treatment. The example, the patient with chronic disease, they will have high chances of non-adherence because they will leave the drug. Because of not giving any effect, they leave the drug. But due to this personalized medicine, the increasing patient adherence also takes place. It also help the overall cost. It also reducing to the cost welfare to the people also. And now we go on to the main important thing, the stages in personalized medicine. We now look on to the clinical research. While we doing a personalized medicine means, first of all, if doing any research means we need a reference, parent reference or parent article. The reference should be very much needed. Likewise, while doing a personalized medicine, the collection of patient and omic data is very important. What patient data should be a demographic age, gender, past history, likewise. What is omic data? Omic data is nothing but, it's a very important thing. Omic data is a collective data, on study on biomolecule. Collective data on biomolecule. The omic data is a collective data of biomolecule. What are the biomolecule data are? Genomic data, which will the DNA sequence takes place. Proteomic data, the protein sequencing takes place, transcriptomic data, the RNA transcript takes place. These are the data is very much important. While we doing the clinical research, collection of patient and omic data is very much important. After collecting data, machine learning stratification. Stratification is nothing but we while collecting the data, we stratification the patient based upon stratification is dividing into their group. What group? This group is a homogeneous group. We see mean it is a homogeneous group. We are similar condition patients should be clustered. For example, we are doing study 100 population under total population under 100 on breast cancer. This is the study. Here stratification takes place, dividing the group into two types. Then the clustering homogenesis, H2 positive groups are one, which are similar condition, and ER positive groups are in another cluster group. And they the using the biomarker, the result will be take place. This is the machine learning stratification. After machine learning stratification, in stages, other stages is translational development. It is also known as preclinical study. Preclinical studies always do on animal, in vitro, in vivo method to accessing efficacy and safety of the treatment through the animal studies takes place. After the animal study, the main picture is came clinical trial. Here the stage based clinical trial have 4 stages, and also phase 0 also takes place. It is also microdosing. While we go to personalized clinical trial, 0 is a microdosing. It should be on less than 8% are positive participate. While we go to the trial phase 1, first in human healthy volunteering takes place is about 8 to 100 takes place. Here we will determine the personalized medicine. In here we determine the maximum tolerability test, genetic testing, profiling biomarker testing. That should be takes place in phase 1. After we move on to phase 2 is the patient, a trial in patient. We will involve the disease similar condition patients in trial, and we also finding the efficacy, safety, and duration, frequency, like things are takes place. In there should be about 80 to 100 population takes place, and phase 3 is a confirmatory trial. It will tell the duration of the treatment and duration of the treatment, when to discontinue, any changes takes place, labeling review, all those things take place in phase 3. Phase 3 also main important thing, FDA approval also takes place, and last phase 4 is post-marketing surveillance. It takes place. This is the clinical trial view of personalized medicine. Personalized medicine in clinical research. While personalized medicine, yearly research should be takes on clear in a research lab only. Why means that should be based upon genetic test, similar condition, similar disease. These are the disease takes place. There's no identification of any biomarker, genetic variation. The proteomic, like omic data are not yearly takes place in much. In the time in Human Genome Project in 2003 is a very important thing in personalized medicine. In 2003, Human Genome Project is completed. That project level revealed the identification of genetic variant and the biomarker, different biomarker. They identified the genetic variant, different biomarker, which help to give a targeted therapy to the certain disease. Certain diseases, and it will help to give the outcome also. The biomarker discovery is the main important thing in Human Genome Project. Till now, there should be a last 2024 FDA approved 8 biomarker. FDA approved 8 biomarker 2004, and FDA approved the 14 drugs in personalized medicine. Then we move to this slide. These slides are takes place in genomic project. After this genomic project, give very important thing. What means early stage should be stuck in a research lab. Now it move on to the clinical setting. Why I am saying this means Now the doctor using the personalized medicine information data for decision making. It acts as a decision making tool also. And however, there is some also drawback also takes place. However, some drawbacks, what are they? Means regulatory challenges. We will see deeply on upcoming slide. Regulatory challenges and some of the challenges will be takes place. We will be seeing upcoming. This is the personalized medicine in clinical research overview. What are the takes place in early research and after the project completion of 2003 Human Genome Project. These are the things that takes place. Clinical trial design in personalized medicine. While we doing a research means we want to make a trial design. Study design is very important while doing the clinical research study. This is a backbone. While we designing the study alone, we will proceed to another thing. Here the type of personalized medicines are divided into 4 types: targeted medicine trial, stratified medicine trial, individualized treatment trial, and individualized dynamic treatment trial. In targeted medicine trial, there are most common 3 trials are biomarker strategy design. Basket trial is very, very important. Umbrella trial. Biomarker strategy design is nothing but we are dividing the people into 2 groups and we starting the treatment, targeted medicine trial to a treatment. And one group they have a biomarker testing, and another group they do not have a biomarker. They do not take place any biomarker testing. And compare the result. Afterwards, we comparing the result. This is the biomarker strategy design. Basket trial. Very important thing in very important thing in personalized medicine. Major studies that takes place in basket trial and umbrella trial. Basket trial is nothing but one drug. Basket trial is nothing but one drug given for different diseases. Example, any one drug given for different diseases. Example, one drug given different diseases: lung cancer, tongue cancer, colon cancer, esophagus cancer. Likewise, different disease. One drug given for different diseases. In this, the same characteristic part means biomarker characteristics are same in these 4 different diseases. This result takes place whether the disease block this biomarker in this disease should be revealed in this basket trial. The basket trial is nothing but one basket have different fruits. Likewise, one basket have different fruits. The one basket is a targeted medicine. Different fruits is a different type of diseases. Umbrella trial is like alternate, vice versa. Umbrella trial. What is mean? That means while doing umbrella trial means in basket trial one drug, in umbrella trial one disease takes place, one disease, and they are divided into subgroups. The subgroups are different treatment. One disease that into subgroups are different treatment. Likewise, the cancer can be divided into 4 or 3 groups as means diseases. If example breast cancer takes place, breast cancer HER2 positive and ER positive takes place. Likewise, we are dividing into subgroups and we giving the treatment, different treatment, and finalize we achieving. Here is a great example. If we have umbrella means inside the umbrella they have a multiple section that is called subgroups takes place. This is mainly used for targeted medicine trial. And next we move to stratified medicine trial. Is major trial is platform trial, but we go form one by one. Classic trial design with subgroup. The classic trial design with subgroup is nothing but classic trial is we know the classic trial. What this means? RTC, randomized trial control trial, RCT. Here we normal randomized studies takes place. In the end of the study, we evaluating the result. That result into 2 types, 2 subgroups. Example, we doing the study on cancer and dividing the subgroup into biomarker testing patient and biomarker not testing patient. We are dividing their group and getting the action, stratifying, dividing the group into getting action. Likewise, I friends say platform trial. Platform trial is nothing but It is a master protocol. It is a master protocol. In another word, they say major trials are takes place master protocol, and it also useful to do many trial in one study. We don't want to move to each trial study. If we choose platform trial means we will done all the study in one trial. How it will be take place mean the multiple treatment given to the certain diseases, certain diseases. If the effective treatment should be continued, ineffective treatment should be dropped out from the trial, and newly added to should be also takes place. After newly added also take place. Example, in COVID-19 there should be a dexamethasone, retinol, antiviral drugs. These are takes place, hydrochloroquine. These are takes place. While we going to this trial, the effective treatment, dexamethasines are effective. That should be placed in the trial. Ineffective treatments are retinol and hydroxychloroquine should be excluded. In any new drug should also added. This it is a continuous trial. It's an ongoing trial. So we need not to perform many trial. It is a single trial give the big answer. That is a platform trial. Umbrella trial I am likewise saying in about targeted medicine. Likewise the umbrella trial also takes place. Then multi-arm multi-stage trial. The multi-arm multi-stage trials are similar to platform trial also. We are dividing the patient into multi-arm. Arm A, arm 1, arm 2, arm 3, arm 4, and giving the treatment, multiple treatment to the drug. Multi-stage, we have a checkpoint in multi-stage, that is the interim checkpoint. While checking interim, there should be ineffective should be drop out, effective should be continue, and new should be added. Likewise the platform trial it's also continued. Marker study is likewise the biomarker study. Above saying biomarker strategy design and marker study base are similar to each other. Then next week you go to individualized treatment trial. The above two trials are based upon groups, one group and two group. This study is mainly based upon individual, each patient. There's no grouping, each patient. That is the classical design separating within and between patient variable. What this means if you taking the patient A, they will give the drug X, example for any drug X, and for 4 weeks response should be noted, and same patient will give the drug Y for 4 weeks response is okay. Their own response should be noted and the result should be published. Individual treatment trial is very less in clinical trial. Why I'm saying means patient recruitment is very low, and due to that the individualized treatment trial is very low. Then move on to individualized dynamic regimen research, sequential multiple randomized trial, SMART trial. It is similarly called as a SMART trial. In SMART trial, what means we are randomizing people. First of all, normal randomized studies take place. Randomizing people into A, B. The A group receive like A drug antidepressant. The B group also receive antidepressant drug. If they after certain week, they should be check the response. If they no response, they again re-randomized. The re-randomize also takes place. Again re-randomize, and the patients are given to, if the patient antidepressant giving any haloperidol given means the same class antipsychotic drug haloperidol risperidone should be given. Same class different drug should be given to monitor the result of the disease. These are the clinical trial research in personalized medicine. This alone takes place in every personalized medicine clinical trial. I cover all those things in this slide is very important thing. Next, role of IRB, institution review board. In personalized institution review board is the main thing. In clinical research, we want to do research in human subject. First of all, we want to give our protocol, investigation brochure, and what are the statement and qualification and participant, and what are the compensation, and what are the reviews should be submitted to IRB to starting pre-year clinical research we want to given to IRB. IRB play major role in the study should be ethically standard. It should be ethically standard, safe to the patient, right and welfare of the patient, safe to the ensure, and it also ensure the right and welfare of the study participants or productor. Any misactivity or misguiding or any discrimination or takes place should be monitored by IRB. And IRB also tell should be will be there in good clinical practice itself. We know when we doing the trial, benefits should be high, rate risk should be low, and it also should be weighted. So safeguard is main important thing. So safeguard and privacy of their genetic data should be evaluated by IRB, and they also access the where will be doing the genetic test also should be accessed, and they also access the counseling of whether the patients should be counseling, the follow up, the regular follow up should be takes place, should be accessed by the IRB. And another one thing is the genetic data is very sensitive because it will release to someone hand means they misuse this. So IRB also give a great access to safeguard the genetic data. While we doing particularly in personalized medicine, the two important boards are main in integrate with IRB. What means National Cancer Institute and National Institute of Health. The National Cancer Institute play major role in providing guidelines for how to create informed consent form. Will be from consent from the patient. How the what are the things are takes place. What are the things should be avoided? Should be guidelines by National Cancer Institute. How the privacy should be protected. How the participant empowerment takes place. How the participant enrollment should be takes place, and how the participant compensation should be given, should be guidelines are given by National Care Institute. In another hand, another important institute is National Institute of Health. The National Institutes of the Health tell the IRB to while we doing a trial in personalized medicine, National Institute of Health tell the IRB to appoint the expertise in genetic should be appointed to evaluate their scientific finding and data should be evaluated. So the National Institute of the Health require IRB to possess the expertise person to appoint to while running personalized medicine should not under the any other person from another related field, should be in from same related field, and there should be a genetic expertise to takes place. These are the IRB role. IRB role is main much important thing in personalized medicine. Then after we move on to IRB, we move on to the challenges. What are the challenges of facing in clinical research? Medical challenges. What are the medical challenges means the drug safety profile is very low because the patient population is also low. So they are not maintaining the drug safety profile in an effective manner. So we want to stronger the drug safety profile to maintain all the hospital formularies should be included, and there should be no how not much larger studies in post marketing surveillance study, post marketing surveillance studies are not much. So these are the challenges, and there should be a in physician the lack of pharmacogenomic knowledge. Lack of pharmacogenomic knowledge. Some of the physicians are have difficult to handle that. There's are takes place, and also while the very few adverse drug reactions should be reported in personalized medicine, there should be unnoted or not filed. So the preventive measures it's very low. So we want these are the challenges in medical types. In legal type, what mean harmonization of law? Why there I previously tell the regulatory challenges are very high. I'm say why say mean the different country have different laws. While US have different law, US FDA have different law while doing personalized medicine. European Union have different law. Canada have different law. So the different law make difficult in doing personalized medicine and production of patient information generator. Because I'm previously tell like the patient information should be generated misused by insurance claimer. They are misusing their information. These are things that takes place. And another thing is biomarker supporting things we want to take place, because while we doing biomarker testing for any rare disease, the clinical diseasing support is lack. So it also need while we testing the patient, it have a lot of lawful regulatory action, like regulatory to follow to check the biomarker test to the patient. These are takes place. And if we doing any biomarker test, it gives you any unexpected result or a patient death, means we will face some lawsuit to us. These are the legal challenges takes place. And next, economical and ethical challenge. Because of this, personalized medicine have big budget, so budget impact takes place. And patient understanding and patient role in future. Sometimes patient refuse to will be going trial. In between they refuse to do something. So it also affect. We also not force them because it is against the IRB rule. We won't force them. So these all takes place. And informed consent form should also use it for long-term use. This informed consent form is not end in this study. It also come back to long term for use. These are the ethical issues. Social challenges. The main social challenges, it is a personalized medicine. So, so what means patient requirement is very low. So very low means the patient requirement is not much enough to do any personalized medicine because of this rare genetic disease, rare thing. So the personal requirement is very low, and likewise funding also low, and there is no standard SOP for hospital and lab. Why means there should be a standard operating procedure should be followed for all the labs and the hospital. They are not following. They are different hospital, different lab follow different SOP. So this is also major problem. And what are the negative aspect is the physician and patient relationship. It also main thing. There is any miscommunication or misunderstanding. It also affect the study. And there are also some positive things are takes place. What means they also improve the quality of healthcare. It also provide effectiveness in study, and public trust also takes place due to the study. Negative also it being point means they're also having high range in positive. These are the challenges, four challenges. Medical challenges, legal challenges. These are the main four challenges. Opportunities of personalized medicine in clinical research. Previously, I'm telling the personalized medicine have major part in cancer disease. They play vital role and major role in cancer diseases. Personalized medicines are personalized now. That is personalized to the oncology department alone. They play major role. They give innovative and initiative trial in oncology department. Likewise, not in oncology also. Nowadays, it also step into another department also. Cardiology, imaging test also takes place for any high risk of CVD, endocrinology, neurology, psychiatric. Personalized medicine also nowadays take place in psychiatric, depression, schizophrenia. Very useful for the patient in depression, schizophrenia, bipolar disorder syndrome patient. Personalized medicines are very important to clinical to get the clinical outcome high rate. It also apply to rare genetic disease disorder also. Personalized medicine takes place, and likewise upcoming year, I'm saying no 2024, the FDA approved 14 drugs, targeted drugs, and 8 biomarker. Likewise, upcoming years also it could be increased 30%. Today, I'm previously tell about the in slide. Likewise, 70% of the targeted innovative drug should be in personalized medicine. 70% of the drug in oncology. It also seemed to be increased. Next 5 years should be 69% of each targeted drug. The innovative drug should be increased 69%, is very useful for life-saving for a cancer patient while we adopt the personalized medicine takes place. From the 2015 to 2020, the number of medicine for personalized medicine is increasing, gradually increasing. After the 2003 research, it gives the way to 2015 to 2020, the long journey. It is a very long journey. It will give the pay to increase 70% of the use of personalized medicine in our clinical setting. And application. Likewise, opportunity itself, I say it, 70% of the innovative drugs are in cancer. How they are using in cancer? Likewise, I previously tell the 6 main stages. Risk assessment takes place, prevention takes place, and detection takes place, and treatment takes place, and management takes place. These are the things take place. Likewise, the personalized medicine, while using genetic tests, they identify the biomarker and genetic mutation variations in people, in cancer patients. That is very useful to, while we testing genetic tests, it result reveal to give the best targeted treatment to the patient. Genetic test give the answer to best targeted treatment to the patient. So the targeted therapy should be achieved through the genetic test. And it also takes place in not targeted therapy. Immunotherapy also nowadays takes place. The personalized medicine used to identify the patient in an individual manner. And after optimizing the drug dosing base, what are the correct optimizing dose, how dose should be given to the patient, it should be individually tailoring takes place. Individually tailoring takes place. Individual trailing takes place, which will also give success rate in treatment outcome and reduce the adverse drug reaction. It also help to identify these are the successful rate, give the weight for benefit of clinical trial. So far alone, these are the successful things. Now the 70% of the innovative drugs are successful in clinical research. This is the major disease application in cancer. Likewise, we move on to next disease, cardiology, cardiovascular diseases. While we giving likewise, the genetic test reveal the high risk of developing patients CVD, cardiovascular disease. So far, we will able to give the personalized treatment to get the optimized drug dosing to reduce the patient from early prevention, and it also reduce the reaction time, and preventing time should also increase in this study. And through that, they also give in preventive lifestyle modification also takes place for patient at high risk patient. The patient receiving have high risk, they should also mainly follow the lifestyle modification plan, behavior plan, and treatment modification plans that take place in patient at high risk. These two things are majorly. And infertility. Infertility is the main disease because a lot of people may be suffer from that. So the personalized medicine tailor for the infertility also. Likewise, genetic test should contribute the what the factor I previously said predisposing factor. Genetic test reveal the what are the predisposing factor, genetic factor, environmental factor, hereditary factor. These are the predisposing factors should be identified, and it also identified the hormonal imbalance also. To this identification, we will tailor the optimized drug dosing for individual. It will give great effect on clinical outcome benefit to the in IVF treatment. For example, if a patient doing IVS process means the hormonal imbalance test and the genetic test will help to modifying their dose or altering their dose, changing the lifestyle, the likewise environment factor. Likewise, it will very helpful, and it also give for personalized plan for couples. It give personalized plan. Due to this personalized treatment plan, give the great achievement result takes place. Last 15 to 20, 2015 to 2020, there is a lot of infertility success rate also in personalized medicine. Then future directivity of personalized medicine. Future directivity, we want to develop the new biomarker because we have alone some new biomarkers are takes place. Likewise, I am saying previously 2014 FDA approved 8 new biomarker. Likewise, innovative of new biomarker will give the pay to different diseases, identify a different disease and identify it very early and helpful for individual treatment very much. Developing new biomarker very helpful for individual treatment and integration of proteomic and genomic data. Likewise, the omic data should be in separated manner, proteomic, genomic, transcriptomic. The integrating of this using AI play major role while integrating this in future because AI will integrate this data in one manner. It will very helpful for the clinical trial purpose. It will analyze the trend and trials while we integrating. That is the uses of data intelligence, artificial intelligence data analysis. Very helpful for clinical trial. Yes, the upcoming future should be based upon artificial intelligence in omic data. And likewise, I'm saying previously, compared to normal trial, in personalized medicine clinical trial have low trial. So we want to do expansion of clinical trial. There have also some challenges due to regulatory challenges likewise. But in future, likewise 2003, give the pathway to the high innovating. Likewise, future also give innovative pathway to the expansion of clinical trial in international level and global data sharing. And in future, there should be, while adapting the global data sharing, likewise, I'm previously saying integration. After integration, the data should be maintained at the, want to create the data bank, that is a biobank, biomolecule bank. Well, the first stages in clinical research I'm saying is collection of data. So it will helpful for researcher to get the database from biobanking. Very helpful to start the clinical trial. It is a stepping stone for first step in clinical trial biobanking. So it also takes place. So these are the things are takes place in personalized medicine and clinical research. Conclusion. We now come to the conclusion part. What means the personalized medicine have a great impact in clinical research. If we tailoring more expansion of clinical trial in more in personalized medicine means it give great impact to clinical research, and due to the clinical research it also give benefit to the patient outcome and improve the patient outcome. Success rate should be increased and effective treatment should be takes place. Likewise, a reduction in trial and error takes place, right rate should be given, and lifestyle modification through adapting the personalized medicine. It will very helpful to for the treatment outcome. And also personalized medicine personalized medicine also accelerating drug development. Likewise, 8 drugs are approved from FDA in last year. In upcoming year 2025, there should be I think there should be 30 drugs are in clinical trial. So upcoming years it should be give approval from FDA, and it also very helpful for the drug to be to give the patient outcome very much, and it also reducing the trial time and cost also. Adapting the technology advance like AI, artificial intelligence in integrating the omic data and data analysis also give great future in personalized medicine. Thank you. Thank you, sir. Thank you for the opportunity.
Thank you. Thank you. Thank you. It was nice presentation. There are
Thank you,
some
sir.
questions, so we will, we'll just take those questions.
Okay, sir.
So Bhavesh has asked you a question. How can we accurately predict an individual's response to a specific drug or therapy based on their unique genetic profile or other biological markers?
Sir, it is a very nice question. While we predicting the individual response, I previously tell genetic test. The genetic test should be subgrouper into genetic profiling and biological marker. Both are takes place. But biomarker nowadays, the biomarker takes place major in identifying the targeted therapy, sir. The biomarker nowadays takes place mainly.
Okay. Thanks. Thank you. Then again, the next question is, what are the most effective ways to tailor drug dosage and treatment regimens for different patients to maximize benefit and minimize adverse effect?
Yes, sir. Likewise, I'm saying in the fourth slide, there should be a six stages. Six stages. First of all, we want to assessment. According to the risk assessment, we will reveal their what their level to takes place. What are their assessment result? Example, any lab result or anything, any result which lead to give a tailoring to individual less drug dosage for the treatment. So we will go to the preventive using a lifestyle modification and treatment. Any treatment intervention takes place based upon the risk assessment. Based upon the risk assessment result, we will tailoring the drug dosage, and it will also
So
give
next,
the.
yeah. So the next question is from Shri. What is the role of biomarkers in personalized medicine?
Sir, biomarker take place mainly used for identifying the targeted therapy. Targeted therapy. If some patient have, likewise I'm saying cancer patient have HER2 positive means using biomarker will exactly target the gene expression, and it also helpful to give the correct target track, and it also avoid the trial and error.
Okay, next one is limited understanding of gene-drug interaction for our population. Is this a challenge in personalized medicine?
Yes, sir. Why means the drug interaction itself very challenging in our India. Some people are not much understanding their drug interaction. They are similarly taking, and they are not much interest in taking knowledge about drug interaction. So gene drug interaction is more larger to drug interaction. The drug interaction should be controlled by using monitoring parameter. But gene direct interaction should be very challenging in personal medicine. It is also one of the challenges. The researchers are work on it, sir.
Okay, so how do we prioritize candidate biomarkers for validation when multiple pathways are involved in disease progression? So this is question from
Sir,
Komal.
how we categorizing the biomarker means depending upon their treatment, sir. The treatment which gene over expression they are affecting, that should be prioritized, and through that alone we validating the candidates, sir, to give the
Okay,
multiple pathway to
okay, and how can AI and big data analytical can be used in clinical research for personalized
uh,
medicine?
yeah, sir. AI is very important. OE means omic data. Omic data is a very much important thing while doing we go to do the clinical trial means omic data is very much important. Due to the omic data alone, that is a stepping stone for the clinical research. For using AI integrating omic data, very helpful for researcher to get the data very much. And also, I'm saying we'll be building the biobanking. AI biobanking means it database means which also help the researcher to get the database very quickly and understanding manner using AI. So AI will play a major role for upcoming future in personalized medicine, sir, for integrating and allotting the database.
There's another question from Harshal. In what way can personalized medicine improve treatment outcome for patients with rare diseases?
Yeah, sir. Personalized medicine also takes place very much in very low clinical trial means trial in rare disease, but it also very helpful for rare disease. Why I'm saying means if we uh, if we if we take the rare disease patient for clinical trial, it will achieve in clinical trial result mean which will help to give the treatment drug to the rare disease patient, and also give success rate also, sir.
Okay. There's one more question. I think this will be the last one. Personalized medicine. How much efficiency shown in genetic disorder diseases? So, is
Yeah,
there
sir.
any information on that? Any genetic disease which has been targeted with personalized medicine?
Sir, basically, the personalized medicine major in oncology, sir. They should 82 percentage of the efficacy takes place in personalized medicine, sir.
Okay. Thank you, Dr. Sriharan. Thank you
Thank you,
all,
sir.
audience, for the questions. And actually, it was very nice session. Simultaneously, it was very interactive. Thank you for your time, everyone. And thank you, Sriharan. Thanks.
Thank you, sir. Thank you for the opportunity.